All right, let's dive in. We're here to run through your mission briefing for BSB1432, the Microbiology Laboratory. So, if you've ever been curious about the invisible world that's literally all around us, well, you are in exactly the right place. So, first things first, let's set the stage. What exactly is microbiology? It's the study of everything microscopic, all those tiny organisms you can't see without some serious help. We're talking about bacteria, viruses, fungi. I mean, it's a whole universe living on us, in us, and all around us. And this lab, this is your passport into that universe.
Here's how we're going to break it all down. First, we'll define your core mission. Then, we'll get into the essential toolkit of skills you're going to master. After that, we'll look at the really cool experiments you'll be running, how your performance gets measured, and finally, we'll talk about why this all really, really matters.
Okay, let's jump right into Section 1, your microbiology mission. This is all about the big picture, the what and the why behind this entire lab course. The official course code is BSB1432, but I want you to think of it more like an expedition. Seriously, this isn't about sitting back and reading a textbook. This is about you getting your hands dirty, metaphorically speaking, and actually engaging with the stuff in a real, practical way. And this right here, this is the heart of your mission. You're here to master the absolute fundamentals, things like handling microbes safely, preparing their food, we call it media, and then inoculation, which is just a fancy word for carefully introducing those microbes to their new home. And the ultimate goal? To be able to pick out one single type of organism from a big messy mix and grow it all by itself. That's a pure culture. Which leads us perfectly into Section 2. Now we're talking about mastering your toolkit. These are the foundational, absolutely non-negotiable skills that every single microbiologist has to know like the back of their hand before they do anything else. And the very first tool in that kit, the one you'll cover in week 1, safety. And let me be clear, this is everything. It's non-negotiable. Before you even think about touching a petri dish, you have to know the rules of the lab. It's about keeping you safe, your classmates safe, and keeping your experiments pure. Everything, and I mean everything, is built on this foundation. All right, next up is probably the most important technique you will learn, period. It's called aseptic technique. Now, it sounds complicated, but it all boils down to one simple idea: preventing contamination. You want to study the microbes you're supposed to be studying, not some random dust bunny that floated by. So you follow these steps every time. Create a sterile workspace, clean your tools, handle everything just right to make sure you're only growing what you want to grow. Okay, so you've got your safety protocols down and you've mastered your basic toolkit. Now it's time for the main event. Section 3, observing the invisible world. This is where you actually put all those new skills to work and start running some key experiments. This timeline is your roadmap for the whole semester, and just look at the journey you're going to take. It builds so logically. You start in week 3 just learning how to see these things with microscopes. Then, in week 5, you learn how to grow them. By week 8, you're basically a detective trying to identify an unknown bacteria. And from there, you're testing how they react to different conditions. It's this amazing step-by-step build-up of your skills and your knowledge. Which, of course, brings us to a pretty important question. How do you know if you're actually getting it right? Let's break down your performance review in Section 4, how your skills are measured. Just take one look at this chart. The message here is crystal clear. A massive 45% of your entire grade comes from your lab reports. This course is all about consistent, hands-on work. And that huge 45% is spread out over 8 different lab reports during the semester. You know what that tells me? This is a marathon, not a sprint. Your success here is going to come from doing steady, consistent work week after week, not from trying to cram everything in at the last minute. Now, let's put it all together. You take that 45% from lab reports and you add the 30% from your practical assessment, and boom, 75% of your total grade comes directly from the work you do in the lab. You just can't pass this course by memorizing flashcards. You have to prove you can do the science and explain what you did. So, what's the target for a successful mission? Simple. The number you need to remember, the number you're aiming for as your final mark, is 40. That's your goal. That's what you're working toward with every single experiment. Okay, so you've done all the experiments, you've written all the reports, you've taken the tests. What's the big takeaway? What's the point of it all? Let's get into our final section, number 5, why this lab matters.
Think of this as your final debrief. By the time you're done, you will be able to do these 4 crucial things. You'll be able to connect the theory from a book to what actually happens on a petri dish. You will be able to walk into a lab and confidently perform basic experiments. You'll know how to use the equipment properly, and maybe, most important of all, you'll be able to look at a set of results and figure out what it's actually telling you. These aren't just bullet points on a slide. These are real transferable skills for your future.
So there you have it. That's the plan, those are the tools, and that's the objective. You officially have your mission briefing. The only question left is, are you ready to gear up and go explore the invisible world?